Hey everyone, welcome to the basics video on membrane transport. Before we talk about getting molecules into and out of cells, let's take a minute to remember how molecules move randomly. This website shows two different molecules separated by a membrane. Before I start, all the orange molecules are on one side and the blue molecules are on another. When the molecules are allowed to move, they'll move from high concentration to low concentration until there is no net difference. I point this out to begin because random motion is a very important force in biology and often misunderstood by students. Molecules don't have motivation. They don't want to move from one side of the membrane to another. Instead, random motion just tends to equilibrate the number of molecules in every part of an area or a solution unless energy is spent to prevent that diffusion from happening. Therefore, the first main factor that affects membrane transport is the concentration gradient of the molecule. If a molecule is diffusing from high concentration to low concentration, it does not require input of energy because passive diffusion takes care of it, and so it is called passive transport. If, on the other hand, a molecule is moving against its concentration gradient, then it does require energy. Another important part of transport is the charge or polarity of a molecule. We've already talked about biological molecules, polar covalent bonds, and the lipid bilayer. It should make sense then that small nonpolar molecules like gases, hydrocarbons, and steroids can cross the lipid bilayer just using passive diffusion. But many important substances are charged or polar, like water, ions, sugars, and small proteins. These can't diffuse across the membrane and must be transported. So let's start talking with passive transport. In these cases, molecules are moving down their concentration gradient. In simple diffusion, molecules cross that don't need a transporter. These tend to be small nonpolar molecules like gases or small lipids. In facilitated diffusion, molecules are large or polar but are still moving down their concentration gradient. They require a channel or carrier protein to get across that nonpolar region of the cell membrane, but they don't require energy. Examples that we've seen before: water, ions, polar molecules. Can you see then that concentration gradient makes both systems passive, but charged molecules require a facilitator to get across the membrane? Active transport is necessary if you're moving molecules up their concentration gradient. We'll focus first on pumps and co-transporters. A pump is a single protein. In this example, it's pumping sodium out, so shown by the circles, and pumping potassium in, shown by the diamonds. In both cases, it's pumping these ions against their concentration gradient. This is a common pump in most cells and requires an energy source like ATP. Co-transport, on the other hand, is a mixed system. It uses a pump to create a concentration gradient, in this case hydrogen molecules, and it requires ATP. Once it has a concentration of hydrogen, it can allow hydrogen to diffuse back into the cell, but only in conjunction with another molecule that the cell needs to move against its concentration gradient. In this case, it co-transports hydrogen with a sucrose molecule. So co-transport involves active pumping plus a facilitated co-diffusion event with the main goal of getting sucrose across the membrane against its concentration gradient. Because ATP is involved, we'll put it under the heading of active transport. A more complicated example of active transport changes the plasma membrane itself. In exocytosis, the cell creates small packets of membrane with molecules inside called vesicles, and we'll see more how this happens later in the class. These vesicles are transported to the plasma membrane, and they fuse with the plasma membrane and release their molecules out of the cell. The membrane that was associated with the vesicle then becomes part of the plasma membrane and makes it just slightly bigger. That's exocytosis. Endocytosis is the reverse of the process, where the cell actually pinches off a section of membrane and brings the membrane vesicle inside the cell, with the desired contents from outside. This allows the cell to use those molecules for its cell purposes, and this, as you might imagine, requires energy. There are 3 different types of endocytosis, which vary in the size of the particles they're trying to bring in and how specific they are. Be sure to pause the video right now, go find this area in your book and write down the definitions of these 3 types of active transport and know an example of each one. Now you've had some definitions in general of transport types, let's look at 3 specific examples that are particularly important in biology. First is the sodium-potassium pump as a mechanism of active transport. This is called an electrogenic pump because it creates a difference in charge across the membrane. The membrane becomes more positive on the outside than the inside. This is an integral protein, which means it sits inside the membrane of all cells, and it runs in a series of, we've defined 6 stages in one cycle of the pump. So let's go through the 6 stages now. In the first stage, 3 sodium ions diffuse from the inside of the cell and bind to the protein. This binding of sodium stimulates the donation of a phosphate group, which is just phosphorus and oxygens from ATP. And the addition of the phosphate group changes the shape of the protein, which reduces the affinity of the protein for sodium ions, which are then released outside the cell. By the way, you'll notice in all of your book's pictures, the outside of the cell is shown as blue and the inside is shown as this peachy color. The new shape has a high affinity for potassium ions, even though it's not very interested in sodium anymore. This causes potassium ions to bind in groups of 2 to the protein. The binding triggers the release of the phosphate group, lets go of the protein, and the loss of the phosphate group triggers another conformational change which exposes the potassium ions to the inside of the cell, and this change reduces the binding sites, the affinity for the protein for potassium, so the potassium diffuses away on the inside of the cell. That's our first example. Our second example is osmosis as facilitated diffusion. And osmosis is defined as diffusion of water molecules in particular. But water molecules act like all other molecules, moving from high concentration to low concentration. As we pointed out, lipid bilayers are not particularly open to water diffusion and require a transport channel. And these transport channels are called aquaporins. Water can move in either direction through these channels, and they move still by random motion like all other types of diffusion. Part of the vocabulary of osmosis are the terms used to describe the fluid around a cell. This is a cartoon of a beaker of saline solution with a cell inside, and this is an example of a hypotonic solution which has a lower concentration of solutes. Hypo meaning lower, tonic we'll use to define as solutes. It's a lower concentration than the inside of the cell. So where is the concentration of water higher, outside or inside the cell? Indeed, the concentration of water is higher outside than inside, and so water will flow from high to low water concentration. It actually doesn't flow across the membrane, it flows through the aquaporins like that. If a cell is in a hypertonic solution, there's a higher solute concentration outside the cell, and so the concentration of water is higher where? Yep, higher concentration of water inside the cell, so water tends to diffuse out of the cell. An isotonic solution has the same solute concentration inside and outside. Iso usually means same. Water continues to flow through aquaporins, but there's no net change, so the flow into and out of the cell will be continuous but equal. Our third example is receptor-mediated endocytosis, another type of active transport. We've talked not very much about cholesterol yet, but it's an amphipathic molecule like the lipoprotein, like the phospholipids that we talked about in the cell membrane. It isn't particularly soluble in water. It's carried in the blood by binding it to proteins, which produce a complex called a low-density lipoprotein. It's got lots of parts to it, which I want you to be aware of. You've got phospholipids and a single layer on the outside. And all these red bits here are cholesterol molecules tucked in amongst the phospholipids and packed in tightly in the center. And then there's also a protein, abbreviated ApoB, that's wrapped around the whole thing, shown in green. And this big protein has a binding site. And the binding site matches the binding site of a receptor that's on the cell membrane. Okay, so a low-density lipoprotein is a collection of proteins and phospholipids and cholesterol that allow cholesterol to be carried successfully in the blood. When an LDL complex binds to the receptor, shown here, it triggers the plasma membrane of the cell to form a pocket. This pinches off and makes a vesicle that enters the cell through the process of endocytosis. This brings LDL and its cholesterol, LDL, into the cell, and the components can then be broken down by enzymes inside the cell, and the cholesterol used for lots of cell processes. This sort of transport provides specificity, since the only thing brought into the cell is something that binds to the receptor, and it's also a mechanism for the cell to bring large items in. This concludes our basics video. Make sure you can answer the following questions before you take the basics quiz. Thanks for watching.